Right. Okay. We started the work off with scientific drawings. Yeah, Albert, you'll need to wait. We're not doing tests, we're doing this revision. So keep up, it won't take long, but we will go through.
Mr. Nice, do we need any books out or anything? Or?
No, you should have notes on this already. Okay, so with scientific drawings, you've got these key ideas. What's a light microscope? What's an electron microscope? How to use a light microscope properly, how to make the adjustments, how to focus on things, parts of the microscope. Teach the rules of scientific drawings. Guys, again, we're not doing the tests. That's something that happens else at another time. Okay, now we won't worry about the Middle Ages and the background. The only thing to take away from those early slides is that people early on knew the environment was a problem in some way, shape or form, but because they couldn't see pathogens and they knew nothing at all about genetics, they just didn't know that you had these disease-causing little things in the environment that could cause problems. With the early microscopes, and this is Hooke's, and he was the one who developed the term cell because he started looking at things like sponges and realised, oh yeah, you've got these regular little structures, and then he started to look at other things under the microscope and he saw regular little structures. So he called them cells after that. Now cells, there's that key idea with cells is that all living things are made up of cells. At least one cell. None of the parts of a cell are alive, but together those parts make up a living thing, a living unit. And for bacteria, it's only got the one, but for you, you've got many. Van Leeuwenhoek, he looked at pond water and things like that, and he was just one of the early pioneers in microscopy. There's your light microscope there. I have highlighted the things that especially you need to know in terms of the names. There's the objectives, and then you've got the ocular lens or the eyepiece up there. You've got the coarse adjustment knob and the fine adjustment knob. Okay. The ocular lens in this case is 10 times, and the objective lenses are 4, 10, 40, and 100 times, and you'll need to be able to calculate magnification.
You just times them together?
Yep, just times them together. So it'll be 10 times 4 is a 40 times magnification. Now, we mentioned before, by way of background, light microscopes use light. Light wavelengths are small for us, but reasonably large compared to other things. And they allow magnifications of about 2,000 times. Yes?
Oh, which PowerPoint is this on?
This is the first one. Okay, on light microscopy. Okay, a light microscope is much like a telescope. Instead, you get smaller images look larger. But the key point there is that it allows a magnification of about 2,000 times or less. An electron microscope, because of the smaller wavelengths, because electrons have smaller wavelengths, and that allows the electron microscope to magnify with much greater power.
Are we supposed to know all the parts of that?
No, that would be much too mean. The key points are here are this. It uses electrons. Electrons have a small wavelength, so you get better magnification. That's it. Now, there are a couple of differences. Okay? Light microscope, whatever we say here on the left is almost the opposite on the right. So this is inexpensive, the other one's expensive. This is, uses visible light and a system of lenses. That uses electrons and a system of lenses. Magnification, 1,000 to 2,000. The electron one can go up to 50,000. So it's much greater resolution, yes?
So do we have to know the difference between a light microscope and
You have to know some basic things. The electron microscope can magnify things more efficiently because it uses electrons. So instead of 1,000 times magnification, you get 50,000 times magnification. Okay, so because of that greater magnification, it provides better resolution and better images. And this last little bit, this is the only one that you can look at living cells here. The other one you cannot, because the specimen's in a vacuum chamber. So you can't put a mouse under there and try and look really closely at it. You can't put anything under there because it has to be, the sample has to be prepared in a very special way before it can actually be viewed. But those are the key differences. That's the same thing, but you can see here, there's a light microscope and there's an electron microscope. Same organism, and approximately the same scale in the images, but much finer detail. So imagine blundering around with a pair of boxing gloves on, and I say, oh look, here are some things on the bench that you have to tell me what they are. And you're sort of going, oh, I think that's a water bottle, yeah? But as things get smaller, it gets harder and harder to tell what they are, because you've got these big clumsy things on. But with your fingers, you can see things at much greater detail, okay, and you can sort of feel things out. And that's key, a key difference between those two. The light microscope, let's see, how to calculate magnification. Just multiply the lenses together. So the ocular lens against the objective lens, and that's it. So you've got this 40 times magnification, 100 and 400 times magnification. As you magnify more, you see less of the object. Yeah? So you can see the whole thing here, but by the end, just a little bit. Yes?
Even though you see less of the object, does it become clearer?
It looks larger, so it becomes clearer for you. Yeah. So that's the trailer. As I said, on the computer, this is much clearer. This is a butterfly wing, and at the higher magnification, you see much more detail. Okay. That's just how to use a light microscope and how to make a wet mount. Those are the many steps of using a light microscope. And there's a little video on how to use the microscope, so that's not a bad thing to have a look at. A few school video on how to use the microscope. The key thing is carrying it carefully. When you use your coarse adjustment knob, look from the side and turn it until the stage is just below the lens, and then you use the fine adjustment knob and make those fine adjustments. Okay. With scientific drawings from the podcast, only use pencil. Okay. Draw 2 to 3 cells. Label lines. Label lines should touch the structure that they're actually labelling. They should be straight, horizontal, and no intersecting. But it seems like we're not testing for horizontal lines this year. So don't tie yourself up in knots trying to get horizontal lines. But that is the convention, that every line is horizontal. Use a descriptive title, very important, and include the magnification somewhere. There's 400 times. Okay, so that's all of the information from the first couple of podcasts and that first slideshow. Now, the cells slideshow is the majority of the work that we have done. Okay, it's large. There's a whole lot of things there, but you don't need to know very many of them. Okay, cytoplasm and, for example, okay, there are your key learning intentions, extensive, and there's more than one page. Okay, so you should have notes, you should have maybe a cram card on those key learning intentions. Those are your key terms. So there's a little bit of revision from year 7. Mrs. Green talks about how we know things are alive, how we classify them, and then it goes from there. Okay, so there's your Mrs. Green. You know, what are the characteristics of living things, and we explored that idea. Yes?
Would you be able to, because I'm still a bit stuck with binary fish and mitosis, would you be able to,
Yeah, we'll come to that, and I'll go through.
yeah.
Okay? So, that'll be the next slideshow. Okay? So there's your Mrs. Green, and you're familiar with that idea that living things, they move, they reproduce, they have metabolism. Okay, that's how we can tell if, how we can tell if they're alive. Okay? But for the year 9 level, the year 8 level, we added this additional information that all living things are made of cells. I mentioned that before. That's key. The smallest structural and functional unit of an organism, all living things are made up of cells. And DNA is a molecule of genetic inheritance and for also producing proteins. So they're not really covered in Mrs. Green, but they are key. You don't get a living thing without cells. Yes?
Does the DNA produce it, or does it have the code to...
The DNA carries the code. The ribosomes construct the protein from that information. So, yeah. So those things are key. Basic requirements for life. Suitable temperature, some nutrients, and water. Without those, you're not getting... you don't have an environment in which life can survive. But life, it could be pretty horrible from our perspective, but it still has to have those things. 100 degrees is not suitable for us, but it is suitable for These are some additional ones. Stable internal environment. That's what organisms attempt to do in any event. Oxygen or other gases for metabolism. And waste removal is key as well. You can't have waste accumulate because that will be toxic. Yes?
What's the difference between the cilia and the flagellum?
Cilia are actually structurally a little bit different, the way they work. Flagella rotate, so they actually have a bearing mechanism, whereas cilia beat. Yeah, Phoebe, you're wasting time. Close your tab. Yeah, just... Okay. Now, let's see. Yes. Multicellular animals. Their living systems, or their lives, are maintained through the coordination of body systems, and we'll talk about these big body systems later, but you won't be tested on those. But we've already explored this idea of, in order to stay alive, you have to have cells in tissues, tissues in organs, organs in organ systems, all cooperating to keep you alive. So it starts at the smallest unit, the cell, but there's a lot of other things going on. That's a really important slide, that one. Don't worry about those little things. All life descended from the first cell, whatever that common ancestor was. It certainly had DNA, but we don't know much more about it. And all life is therefore related. You have two types of cells, prokaryotic, eukaryotic. Eukaryotic means true nucleus. Bacteria and archaea, these are the domains of life at the top. Bacteria and archaea are prokaryotic, and eukaryotic cells are everything else. And for us, the key things in terms of, I'll look at the kingdoms. We have those two types of cells, we have those three domains, and we'll look at the kingdoms in a little bit, but there are those 6 main kingdoms that you have to be familiar with. We know what a cell is, smallest structural and functional unit of a living thing. Prokaryotic cells don't have organelles. They're very small, they're 1,000 times smaller, so you need to know a little bit about sizes. They're 1,000 times smaller, 10 times shorter, 10 times less wide, 10 times less high. And so everything's bunched together and nice and close. And so they don't have a true nucleus, they don't have a membrane enclosing DNA, they just have their genome, their chromosome just sitting there. And whereas eukaryotic cells, it does mean true nucleus, and they have organelles in them. And you need to know a few of those organelles. Okay? Let's see. And the eukaryotic cells are significantly larger. So that's a key difference between them. Now, we talked about surface area to volume ratio, and you should understand that idea, that the smaller the cell, the greater surface area to volume ratio. And we even did the math, where we had a single unit along each side, had a surface area to volume ratio of 6, but for the larger eukaryotic cell, 3 by 3 by 3 sides means that the surface area is 54 in total, but the volume is 27, and when you do that equation, you end up with a surface area to volume ratio of just 2. And why is that significant? It's because if you're a very small cell, it's easy to transport nutrients in and to get waste out. But if you're a very large cell, it's because your volume inside is so much larger, you need these organelles to keep everything in place. And some of those structures, like little vacuoles, are used for transporting stuff out of the cell and even for transporting stuff in the cell. So you need all these specialised processes to go about living. Okay? So eukaryotic cells can be larger because they contain all of the key processes inside organelles. So the organelles keep all of those processes local in this tight little area, and then the rest of the cell is doing other things. But what that organelle is doing is a particular job. There are your 6 kingdoms. Okay. So a single cell is obviously made up of one cell, and they're prokaryotic and eukaryotic. You get single-celled eukaryotes. Multicellular organisms are always eukaryotic, never prokaryotic. No bacteria or archaea are multicelled. They contain many cells which are specialised in different types of tissues. There are the 6 kingdoms. The prokaryotes are archaea and bacteria. The eukaryotes are animals, plants, fungi, and protists or protista. And you do need to know those 6 kingdoms. Let's see. And you can see in this our tree of life here, the bacteria were the first of the kingdoms, and from those there was that split at that point. Then you had archaea or archaeobacteria, but it's better to just put archaea. And then protista, plants, fungi, and animals sometimes later. Now, this is starting to get a little busy, and the next few slides look at the key organelles that you need to be familiar with. So eukaryote means true nucleus, and plant and animal cells have some similarities. I've put red dots near the things that you need to know. So that you can see there are many other words, but you just need to look at those red dots. Okay? And what do they mean? Organelle is a membrane-bound compartment, okay, which are within eukaryotes, and they have specialised functions. So the first one is the cell membrane, which surrounds the cell. It keeps the cell things inside, and it keeps the environment outside, and it sort of it maintains that internal environment. And there's an element of selectivity. Not everything gets in, and not everything gets out. So it's a semi-permeable barrier which limits what can enter the cell and limits what can exit the cell. Yes?
What's the difference between cytoplasm and plasma membrane?
I'll do that. Okay, there's cytosol and cytoplasm. Okay, so cytosol is just that mainly water, okay, environment of the inner cell. The cytoplasm is the cytosol and the organelles together. And sorry, what was your question?
What's the difference between cytoplasm and the plasma membrane?
The membrane is the membrane. The cytoplasm is the liquid with organelles in it. Yeah. So yeah, are you asking this for the cell membrane and a plasma membrane?
Yeah, a little bit.
Because there's cell membrane up here. Cell membrane and plasma membrane would be functionally the same. Yeah. But cytoplasm is liquid with it's cytosol plus organelles, and cytosol is just liquid. Yes.
Do we have to know all these definitions?
Yes.
Yeah.
You have to know what these key organelles are.
Yep.
Okay. So the nucleus contains the DNA, and that's the control center of the cell. It controls the cell's metabolism, and it controls, in terms of hereditary, what gets passed on, because that's what DNA does. Bacterial chromosome, there is no membrane around it. There's no true nucleus. It just floats there in the cytoplasm. Now, ribosome, you aren't required to know. Okay? That's why it's pink. Flagellum, you aren't required to know. It's a boundary thing, because I have seen some questions where they mention ribosome as a wrong option in multiple choice, if you like, just to throw people off. So the ribosome's important because that's what makes proteins. DNA carries the instructions, ribosomes make some proteins, and you are proteins. Yes?
Do we need to know that process of glucose into the ATP?
Yes.
Okay.
That's cellular respiration, which is the fourth slideshow. And I said, yeah, you'll need to know that. Flagellum is just that interesting little whippy thing. You can have one or many. That's for moving, for cellular movement. Okay, interesting, but again, not something that you can really be tested on. Mitochondria, you can be tested on. They produce energy through cellular respiration, which is the fourth slideshow. And you need to know that that's the organelle that does that. That's what's keeping you alive. Yes?
Could we stay on the heels?
Yeah, I'll grab that in a little bit. Okay. Now, cellular respiration produces ATP. There's a slideshow on that. Okay. Now, those previous organelles are common to plant and animal cells. Plant cells alone have these green ones, and that's a key difference. So if you see a picture and you recognise a large vacuole or a chloroplast, so have a look at those. Let's have a little bit of a squeeze. You made some posters on this. There's your chloroplast over there. It's got these little green stacks in there. The mitochondria kind of looks like a Mexican bean. They've done it in blue. Normally, a mitochondria will be something like they'll do it like that. So it looks like a clan or a little Mexican bean shape, whereas a chloroplast is slightly different. Okay, but cell wall is important for plant cells. It gives them strength because they're bunched together like bricks. Okay, a large vacuole is holds primarily water. It can hold other things. It's found in the centre of plant cells, and when it's full of water, it makes the cell. It pushes everything up against the cell wall and gives the cell a nice plump structure and gives it rigidity. If the vacuole runs out of water, the cell starts to go all soppy and deflate. Chloroplasts, key again, photosynthesizing organelles, and you do need to know photosynthesis, organelles of eukaryotic plants and some other protists. And that's where photosynthesis occurs, the conversion of inorganic nutrients into organic glucose. And that's it there. That's the equation you need to know. Carbon dioxide, that's both a word equation and a chemical equation. Carbon dioxide and water through light and chlorophyll produces glucose and oxygen as waste products. Okay, you will need to know that. So you need the photosynthesis. This is blue stuff as a bit of background. Okay, all plants on Earth incorporate symbiotic cyanobacteria, which are chloroplasts, to do their photosynthesis. So it's literally chloroplasts are an symbiotic bacteria that lives inside plant cells. Okay, so those bacteria, started off in the ocean 3.5 billion years ago, been quietly photosynthesizing and changing the environment ever since. And one way they got out of the ocean was to do that inside the cells of other organisms. Okay, and so yes, all plants, all photosynthesizing plants are green because of those symbiotic cyanobacteria. There's a little comparison between animal plants, fungi and bacterial cells. It's quite useful. In a lot of cases, with the eukaryotes, there's going to be similarities all the way down, but of course with animal cells or plant cells, they have those three key differences that distinguish them from animal cells. Fungi, I've not seen a fungi question pop up recently, but it can happen. And bacteria are obviously distinctly different. Okay. Now, and there's a little picture there. Now, where are we? There we go. Okay, cell division was what we did earlier in the week. Okay, and this is where we got binary fission versus mitosis. There's a good video on this, it's worth watching. Okay, the key thing with asexual reproduction, single-celled organisms, you can have multi-celled, but we'll deal with single-celled. No. The offspring are identical to the parent cell. No difference.
Is that
Whereas
asexual?
Sorry?
Is that asexual?
That's asexual, yes. Because you've got one cell making a copy of itself, the organism is identical. In sexual reproduction, the offspring is a combination of the parents, and so it is not identical to either of them. Okay? Key learning intentions are there. You need to know what is sexual and asexual reproduction. Now, let's see. You need to know a little bit about binary fission versus mitosis. Yes? Yes. Discuss that mitosis is a type of cell division for growth and repair. Okay? Versus binary fission, which is for reproduction. And let's see. There are those key terms again. Now, DNA, we've talked about that before, carries genetic information. It's responsible for passing that between generations. It's also responsible for proteins via the ribosomes. Okay, reproduction can be sexual or asexual. The parent DNA is passed on to the offspring. Okay, now, okay, sexual reproduction is the combination of cells from two eukaryotic multicellular individuals. It's eukaryotic, it's multicellular. Okay, it's not ever prokaryotic, and it's not ever single-celled for sexual reproduction. Okay, let's see. It takes time, a little bit more energy, but you get a genetic blend. Prokaryotic cell, asexual reproduction can be prokaryotic or eukaryotic, but it's always single-celled. Okay, it's only one parent is needed. The offspring are produced quickly, but you have a clone of the parent, absolutely identical. So if a disease comes through, and the immune systems of the clones will all be the same, and so they'll be equally likely to die. Whereas genetic variation from sexual reproduction makes everything a little bit different. Yes?
So asexual is just like a unicellular,
Yes.
just one
Yeah.
with
Single-celled,
prokaryotic.
yeah, single-celled prokaryote or eukaryote, because fungi, single-celled fungi will use binary fission as well. And there's binary fission. You need to know those key differences. Yes, the cell is divided in half, but what are the steps of that process? Make a copy of the chromosome. The chromosome copies are attached to the ends of the cells. As the cell gets longer, those copies are pulled away from each other, and then you simply have the cell membrane and wall, if it's relevant, work its way down and just pinch off, and you've got your two identical cells. Okay, so same thing we mentioned before, prokaryotes and eukaryotic yeasts use binary fission to replicate. Now, mitosis, what are the main things happening with mitosis? Growth and repair in eukaryotic organisms. We don't do, the process of sexual reproduction occurs through meiosis, which is done in year 10. Okay, so this is key, growth and repair. The other one was for reproduction. Okay, again, you get copies being made. Okay, now, but this is not for in terms of sexual reproduction. We have a cell here. Its nuclear membrane dissolves. It makes a copy of each chromosome. The chromosomes light up in the middle, and these spindle fibres attach to the chromosomes, the centre of the chromosomes, the centromere it's called, and then they just reel them in and pull the chromosomes to the ends of the cells, and then you get a nuclear envelope forming again, and finally it actually will splice down the middle, and you'll get yourself a second cell at that point. And that's what's happening in you all the time, every day. That's how you keep replenishing cells that are killed off or die from old age. And finally, cellular respiration. And we mentioned this before. We mentioned certainly photosynthesis just before. 6 carbon dioxide, 6 water, light, and what's the thing that goes under the light? Chloroplast, that's where it's occurring. Produces glucose and oxygen. Now, of course, cellular respiration is the flip side of this. You start with glucose and oxygen, the same amounts. Through mitochondria, you get 6 carbon dioxide, 6 water, which we started with down here, and ATP energy. Okay, is the thing that we're after. So plants do this all the time. They make their glucose and then they use the glucose. But we as animals can't make our own glucose, so we have to get it from other sources, either vegetable material or animal material of things that we eat. Okay, so finally those last key learning intentions, you'll obviously need to know what aerobic cellular respiration is. We say aerobic because it needs oxygen. There's other forms of cellular respiration, but we at this level are just interested in those that require oxygen. And this is why when you're running, you need to breathe more rapidly, because the mitochondria, the ATP that they're producing, they're having to produce more of it as fast as they can. And they need oxygen for that. Yes?
Is the bacteria down the bottom right corner, does that have cilia and flagella?
No, it's got flagella, and I don't even know if that's accurate. It certainly has flagella. I don't imagine it's supposed to be cilia. I think it's just supposed to look cool. Okay. Now, cells, okay, you already know what this is. Those specialised single units of life that all living things contain. For us, multicellular, we have many different types of cells in our body. A bacteria is just one cell. It's only got a couple of jobs. Okay, make a bit of energy, find nutrients, utilize them, make a bit of energy, and every so often, make a copy of yourself. Yeah? So it doesn't really have a lot to do. But these cells, the many types of cells that make up us, they have their primary job to do, and they work cooperatively to keep us alive. Cells are grouped in tissues, tissues are grouped in organs, organs are grouped in organ systems. All those organ systems working together keep you alive, keep you ticking. And because of all of this cooperation and communication, you have high energy requirements. Okay? And that's what's covered on the next slide. So prokaryotic cells, because they're small, they're simple, have simple metabolisms. They make proteins, they reproduce through binary fission, and they might move. A lot of bacteria don't. They just sort of go with the flow of things. They just go where the water takes them. Eukaryotic cells, they're large, they're complex, metabolisms require lots of energy. They also make proteins. They also make copies of themselves through mitosis, growth and repair. But they also have specialised functions and to maintain the health of the organism, and because of that, a lot of energy is needed, more energy. So we do aerobic cellular respiration, bacteria don't. They don't need to. Bacteria and archaea do not need to do that, but we do in order to survive. And here again are the chloroplasts and the mitochondria. We reviewed this earlier. In chloroplasts, they photosynthesize. They take carbon dioxide and water, and through sunlight, convert that to glucose, and that glucose is then used in the mitochondria through aerobic cellular respiration. Oxygen and glucose go into the mitochondria, and ATP is the energy product, and carbon dioxide and water are the waste product, and they can feed right back into photosynthesis to be used again, and it goes from there. So you need to know which organelles do that, and you need to know the chemical equations. Okay? Let's see. Now that's the cellular respiration is defined as the oxygen requiring process which converts glucose from chemical food energy, converts glucose chemical food energy into the energy of the ATP molecule. And that equation is right down there, both as a word equation and as